Bioinformática en the Comunidad Científica Lambayecana. My name is Marco Mechang. I'm a postdoctoral researcher at Michigan State University, and today I'm going to moderate this seminar. Today we are honored to host Dr. Joae Setubal from the University of Sao Paulo in Brazil. But before we start this seminar, I want to tell you a little bit about the organizers. The Sociedad Peruana de Bioinformática, or the Peruvian Society of Bioinformatics, was created on July 23rd, 2019, and this society aims to integrate the Peruvian community interested in bioinformatics and also wants to promote the development of this resource area throughout the country. And the Comunidad Científica Lambayecana was created in 2017, and its mission is to integrate scientists to promote research in northern Peru. So please, everyone, turn off your cameras and your microphones to avoid any issues with the during the presentation. And if you have any questions, just write it down on the chat box, and I will read them all at the end of the presentation. And just to avoid any misunderstanding of misinterpretation of what you want to ask, if you write the question in English, I will read it in English. If you write the question in Spanish, I will just read it in Spanish. So as I said, we are honored to have Dr. Joae Setubal from the University of Sao Paulo, and his research focuses on metagenomics, phylogenetics, and horizontal gene transfer. And he also works on the comparative genomics of the plant pathogen Xanthomonas citrei. And today he's going to tell us about bioinformatics, genomics, and coronavirus. So please help me to welcome Dr. Joae Setubal. So now you can share your screen.
Okay. Let's see if the host disabled participant screen sharing.
Oh.
Uh-oh.
Okay, sorry. You can do it now.
Okay, let's try. Okay. Can you see my screen?
Yes.
Excellent. Okay, so first of all, buenos días, buenas tardes, buenas noches. Good morning, good afternoon, good evening. Thank you so much for the invitation to give this talk. I was told by Marco and Percy that there might be people listening from different parts of the world, which might be at different time zones. I know for sure that we have 3 time zones here. My own, which is in Sao Paulo, Brazil, and Marco who's in Michigan, 1 hour behind, and Percy in Peru, which is who is 2 hours behind. So in any case, hello to everyone, and thank you for the invitation to give this talk. Before I begin, I would like to mention that this is the first time that I'm giving an online talk. And I wasn't very sure about the content. I wasn't very sure about who was going to attend. Percy did give me some information about the audience, but I made the decision to present a very simple talk as opposed to a complicated research talk, which means that everyone who's in the audience who is works with bioinformatics and genomics will find that the first half of the talk to be very simple. It's basically bioinformatics 101 class as opposed to a research talk, but I felt that I had to make this choice because I thought that it would be the most inclusive given the information I had. So I apologize in advance for for whom the first half of the talk is trivial. And hopefully, if you stay long enough for the second half, which we'll talk about coronavirus, it's going to be a little more interesting. Okay, so I will start by this question of what is bioinformatics? And I like to start with this question because different people have different ideas of what bioinformatics really is. And my own view is that, first of all, bioinformatics is the same thing as computational biology. I know that there are a lot of people who think that bioinformatics and computational biology are two different things, but I have never seen a consensus as to the different meanings of these two concepts. And given that there's no consensus, I think that the easiest way is simply say that, well, it's the same thing. And what is this thing? This thing is the most concise definition that I can come up with is the one that you're seeing, which is development and use of techniques and methods from the exact sciences to study and understand biological phenomena. I think this definition is broad enough to basically encompass basically all activity that goes on in bioinformatics. But in my case, I like to restrict this meaning to molecular biology as opposed to the whole of biology, and even more especially to genomics and omics in general. And I make these restrictions not to say that bioinformatics only applies to molecular biology or genomics, but simply to say that my activity in bioinformatics is restricted under those two headings. And historically, it is true that bioinformatics started out as being recognized as a field primarily because of genomics. And the word omics also requires some explanation. Everybody's familiar with genomics, but there's also transcriptomics, proteomics, metabolomics, lipidomics, and so on and so forth. I think basically any object of interest in molecular biology will have an omics associated with it, as long as you can measure or obtain data related to that, and especially large volumes of data, which allows me to say that a feature that is shared by all these omics is that they have large amounts of data. And so they follow under the general heading of big data applications. And precisely because you have large volumes of data, you need computational expertise, computational infrastructure in order to make sense of all these data. And that's basically another way of expressing the concept of bioinformatics or computational biology. And I also like to emphasize that we are going through a genomic revolution because of the tremendous technological evolution in terms of DNA sequencing machines that has taken place in the last 30 years. And that revolution is best seen by two facts. The first one is the exponentially increasing throughput. In other words, the number of base pairs that a given sequencing machine can generate per time unit. And the second fact is the exponentially decreasing costs. And that basically means how much does it cost to generate DNA reads. And the fact that they are exponentially increasing and decreasing, those two features, is simply extraordinary. Very few, if any, other areas in technology have gone through this evolution this increase in throughput or this decrease in costs. And it started basically in the early 90s, and it's still going on, and it's not clear when it's going to stop. At some point, it has to stop, but I'm not aware of any predictions that it's going to stop anytime soon being the next few years. And it's also customary to show graphs that depict this increase and this decrease. And so this graph that you're seeing here shows the exponential increase in throughput. On the y-axis, you have those numbers, which are kilobases per day per machine. It's a log scale, which means that a straight line would already be an exponential increase, and you can see that it's more than linear. So it's super exponential, in fact. And in the x-axis, you have time. And even though this was published several years ago, the message is basically the same. And the second graph is the cost, which I said was decreasing. And again, you have here the cost on the y-axis, also a log scale, and the x-axis you have time. And you can also see that it's super exponential because the decrease is not linear, and it's really extraordinary. All this technological innovation has had tremendous consequences, and one of them being that genomics has spread in many biological areas as an important tool. Many areas which 30 years ago wouldn't even think about using genomics for anything. Nowadays, genomics has become an essential tool. And I will mention a few initiatives that are current that show the expansion of genomics thanks to that technological revolution. The first one is an announcement by the United Kingdom's Department of Health and Social Care. About a year and a half ago, they announced that they were going to sequence 5 million human genomes over the next 5 years. And you know, for a Department of Health to announce that 5 million genomes will be sequenced is a major initiative. It's a national initiative, and it's only one of many. Another example that I wanted to share with you is this other initiative called the Earth BioGenome Project. This is an international initiative spearheaded by Harris Lewin from University of California in Davis in the US. And he and a bunch of other scientists published this paper in PNAS in March 2018, also about a little more than 2 years ago. And basically what this paper states is that they think it's worthwhile to sequence, catalog, and characterize the genomes of all of Earth's eukaryotic biodiversity in 10 years. So this is a major project, a major initiative. When you think that eukaryotes on Earth are a very diverse group, as you can see by this cartoon of containing turtles and whales and plants and carnivores and fish and so on and so forth. So it's 10 years ago, nobody in their right mind would propose such a project. But after all this revolution that I mentioned to you, now, people will look at that and say, hey, great, do you have the money to do that? If you do, by all means, do it. So that's the typical reaction. of course, getting the money to do that, even though the decrease in costs has been exponential, getting the money to do that is still an issue, a major issue, and this PNAS paper is in part a way of the authors or of the leaders of this initiative to convince agencies that it's a worthwhile effort. The third example is a project that has a name similar to the previous one. The previous one was the Earth BioGenome Project, and this one is called the Earth Microbiome Project. It's an initiative that has been going on longer, and I think it's less well organized than the EBP, but even though it's despite that, it's ongoing, and the aim is to sample many diverse habitats on Earth in order to learn about the microbial communities that live in those habitats. And it's, again, also a very major initiative, and its existence is due to the genomics revolution. Okay, with that as motivation, I will, over the next few slides, make some explanations as to what genomics means and what role does bioinformatics play in genomics. And I'll start out by saying that genome sequencing means only that someone will generate a collection of DNA fragments. And with those fragments, there's not much you can do. And in order to have that collection of DNA fragments to be useful, there are several steps that need to be undertaken. And most of those steps are made possible thanks to bioinformatics. Many of those steps are pure bioinformatics. Until you generate the DNA reads, that was made possible thanks to technological innovations in biochemistry and in many other fields. But once those fragments have been generated, then bioinformatics takes center stage. And the very first step generally is fragment assembly. Those fragments, in the case of isolates, they come from specific organisms, and you want to know the entire sequence of that organism's genome. And in order to do that, you have to do genome assembly. And in this slide, you can see a very simple description of what genome assembly means. So there are a couple of concepts here. You have the fragments, which are the blue segments on the top. And then you have this bar, this blue bar here, which means that somehow you were able to join these fragments into a larger fragment. And this larger fragment gets the name of contig. And the way that the fragments can be joined can be the easiest way to explain it is by this the lower part of the slide, where you can see that I have the actual sequence of a fragment on the top and the actual sequence of another fragment at the bottom. And it turns out that the end of the top sequence is the same as the beginning of the bottom sequence. And when you align them, you can see that they match. And this matching, assuming it's not a random match, it has a biological meaning. It simply means that those two fragments came from adjacent regions of the genome. And if they did, then you can join them. And now you have a fragment which is larger than the two component fragments isolated. And by doing this many times, looking at all the fragments, then you will get ever larger fragments or ever larger contigs. And in the best case, in the case of a bacteria, for example, that has a single chromosome, you will end up with just one contig, which represents the chromosome of that bacteria. More generally, you are not able to reach that point, and that's what I describe here, in which you can see that you have two separate contigs, and they are separated by a gap. And the gap exists because when you did the sequencing, you were not able to cover a piece of the genome. No fragment was sequenced for that piece. And if that happened, then you will have a gap. And it's true that pretty much all eukaryote genomes that have been sequenced have gaps. In spite of all that technological revolution that I described to you a few slides ago, it is true, it's still true that nearly all eukaryotic genomes have gaps, even like fungal genomes. And the reason is that it's still not cost effective to close the gaps. We do have the technology to close gaps, but the cost for closing the gaps in relation to the cost of obtaining the reads has become very large. And because most people feel that they can learn basically everything that can be learned from a genome by this kind of draft sequencing. This means that most genomes, nearly all genomes, are left with those gaps. And therefore, they are considered draft genomes as opposed to finished genomes. In the case of prokaryotes, bacteria and archaea, then there are lots and lots of genomes that have been finished and have no gaps. But even there, it's still the case that the majority of genomes that are available do have gaps. Okay. After genomes have been assembled, they still are not very useful because assembled genomes, they simply mean a collection of A, C's, T's, and G's. And the interest in genomes exists because they contain information. They contain genetic information. And where is this genetic information? And in order to, in the process whereby you attach meaning to a genome sequence is the process of genome annotation. And when we talk about genome annotation, we have to talk about genes because the genes are the main unit of information in any genome. And you can see there this figure that shows the concept of a gene. Gene at the barest concept of a gene is simply a section of the DNA molecule. But it's not any section, it's a special section. It's the section that does contain information. And in what kind of information? Well, it depends on the type of gene. You have protein coding genes and you have RNA genes. You have transfer RNA, ribosomal RNA, long non-coding RNAs, and there's a long catalog of RNA genes. And in the case of genes from bacteria and archaea, they are simpler than genes from eukaryotes. They have a structure which is described in the figure that you see. There are components. You have the coding region, you have the ribosomal binding site, you have the promoter region, and so on and so forth. And the structural annotation, what some people call structural annotation, is the process whereby you find where the genes are and where are its components. After that comes functional annotation. And functional annotation is the process whereby you attach functional information. Genes are the protein coding genes. The proteins have functions in their organisms. And you want to know what function do these proteins have? So here you have a very simple example of the kind of information that I'm talking about when we talk about function annotation. And in this case, the organism is Escherichia coli, E. coli, strain K12. And this is information about one of its genes. E. coli has about 4,000 genes. And one of them is the famous LAC-Z gene. And somebody annotated the E. coli genome many years ago. And for LAC-Z, what you see is basically what the kind of information that was attached to the genome sequence thanks to processing of the genome information in addition to lab experiments. So you have the gene name, you have the protein name, you have the location, which came from the structure annotation. You know the protein length, which in this case is about 1,000 amino acids. And most important of all, you have the function. LAC-Z has this function, hydrolysis of terminal non-reducing beta-D galactose residues in beta-D galactosides. So if you know your biochemistry, this will make sense to you. If you don't, well, sorry, but that's the function. And how do we know that this gene has this function? Well, that comes from, in this case, from lab experiments, and these lab experiments have been published. And so ideally, in the genome annotation, you have pointers to the literature where you will be able to understand how this conclusion was reached. Okay, suppose then the genome was sequenced, it was assembled, it was annotated. What happens next? Usually, annotated genomes have to be made available to the scientific community. And in genomics, the most important database of genomes is the one maintained by NCBI, the National Center for Biotechnology Information. In addition to NCBI, there are lots of other sites where you can find genomic information. And some of these sites offer tools for genome analysis. And one very special type of genome analysis tools is the ones that allow comparative genomics. Thanks to the genomics revolution, nowadays there are thousands upon thousands of genomes available. And one very powerful tool is a tool that will allow you to compare different genomes either from related organisms or from distantly related organisms. So for example, in the case of bacteria, so Marco mentioned that I have worked with Xanthomonas bacteria, which are plant pathogens. So we can learn a lot about the Xanthomonas genus if we have the genomes for different species of Xanthomonas. We can if we know about their phenotypes, that this strain, this species of Xanthomonas causes disease in orange, and this other species of Xanthomonas causes disease in rice, then by comparing their two genomes, we may learn important information as to why one is specialized in oranges and the other is specialized in rice. Okay, and well, how can all that serve a purpose? And I would like now to switch gears to coronavirus, which is something that became a worldwide concern, as all of you surely know. And this, my choice of coronavirus was motivated by the fact that we are through this pandemic. But it also, being a virus, it's easier to explain compared to other more complicated organisms. I will now start talking about the genomics of coronavirus. But I have to say that this is not my research field. I am not a coronavirus researcher, but there's plenty of information available nowadays about coronavirus. And I found in previous talks that people like to hear about what's being done with coronavirus, and so I thought this could be useful for this talk here. Okay, so let's talk about coronavirus genomics. Here you have a microphotograph of coronavirus showing cross sections of the viruses with their typical crown-like appearance, hence their name. I mentioned before that there are sites where you can find genomic information. In the case of this talk, one of the sites that I used has this name and URL that you can see there, the Virus Pathogen Resource. It's a very useful site. Anyone interested in coronavirus genomics should take a look at this site. Here's the homepage. It's a site that not only contains many genome sequences of coronaviruses, it contains sequences from all kinds of viruses. At the bottom, you have here a list of some of the viruses that they host. On the left, you can see that there's this class of viruses called single-stranded positive sense RNA viruses. And coronavirus is in this class, which means that it's an RNA virus, it's not a DNA virus, and it means that it's a positive sense, meaning that its RNA is ready for translation. And in the middle, you have single-stranded negative sense RNA, another group of viruses. Then you have double-stranded RNA viruses, and finally, you have double-stranded RNA viruses. And in each of these categories, there will be lots and lots of different viruses. And at the top, you can see that this site offers you tools to search for sequences or epitopes and host factor data and antiviral drugs. It allows you to analyze sequences. So you have sequence alignment, phylogenetic trees, and so on. And you also have what they call a workbench, meaning that this is a private space of users that register in this site. This is very useful for people who are working with specific viruses. And this is a picture of the coronavirus structure that I took from this site. And you can see some of the main features of the virus. In the middle, you have the RNA genome. On the outside, you have the spike glycoprotein, which gives the virus its appearance. At the bottom here, you have a representation of the coronavirus genome, and we'll see additional representations later on, so I don't want to spend too much time on this representation. I prefer to concentrate on the ones that will show up soon. Here you have another picture of the coronavirus, which must be familiar to everyone by now. In red is a representation of the spike protein. Of course, this is not a photograph. This is computer graphics representation. Here's a list of features of the coronavirus genome that I took from that site. Probably I should simply go ahead and show you this graph. I made this graph based on data that I got from GenBank, and it shows the number of SARS-CoV-2 genome sequences, complete sequences that have been deposited at GenBank over time. So on the x-axis, you have time measured in days. The first point here is March 15th, and the last point is last week, May 29th. And on the y-axis, you have number of genome sequences. So back on March 15th, there were just something like less than 100 sequences available. And you can see that it quickly went up. And it looks like starting in early May, the rate of new sequences available seems to be decreasing. But that's already over 3,000 sequences, and that's a lot of sequences for a virus. The very first sequence that was deposited was the famous Wuhan, which was the one from Wuhan in China. It was sampled in December 2019. And here I have a screenshot of the NCBI SARS-CoV-2 resources, which is another site where you can get information about coronavirus genome sequences. It's a little more user-friendly than the one from the previous site. I was very quickly able to list all coronavirus genomes that have been deposited in GenBank that came from South America. And there are 6 genomes available at GenBank as of, I think was last Monday, the day before yesterday. And you can see that there is 1 sequence from Peru, 2 from Brazil, 2 from Colombia, and 1 from Uruguay. So these are just complete sequences, complete genome sequences. I know that there are lots of additional sequences coming from South America, but either these other sequences are not complete genomes or they have not been deposited at GenBank. At GenBank, as of 2 days ago, just these 6 sequences. Okay. So here you have another representation of the coronavirus genome. And now you can see a breakdown of its genes. On the top here, you have the first open reading frame, which is called ORF1AB. It actually codes for several separate proteins, but they are transcribed all together. That's why you see just one ORF here. And then in blue, you have additional ORFs. And S is the spike protein that I mentioned before. And then you have additional ones here. As you can see, it's a small genome. It has about 30,000 base pairs. This, it's in a certain sense, in an anthropocentric sense, there is a view from humans, 30,000 base pairs is almost nothing because we humans have 3 billion base pairs. And yet, you can see the damage that this little thing is causing. I think you gain a deep respect, even being so small. Okay, now in this part of the talk, I will go through a quick description of the components of this genome. And in order to present you with this description, I am using an article that was published in the New York Times last April, last month, and it was a very good article. Its authors are these journalists, Jonathan Karam and Carl Zimmer. I have here the URL in case someone wants to check it out. But it was very well done, and it made my task of presenting the genome of the coronavirus much easier. So first of all, here you have the another description of the genome. Let's see here. Here you can see, you know, at the top you have the ORF1ab and then the structural proteins. Here's the spike protein, and here you see the components of the non-structural proteins that are part of the ORF1ab protein. You have 1, 2, 3, all the way to 16. And in the next few slides, I'm going to show you examples of what these genes are. So starting with the very first one, NSP1. And here you have a representation of its structure once the protein folds in 3 dimensions. This representation is the one that seeks to be the closest possible to the way the protein actually looks like. Of course, the colors are all made up colors, but the little bumps that you see here are representations of the atoms. And so what does this protein do? You have here a description. The protein slows down the infected cell's production of its own proteins. And therefore, the journalists have described it as a cellular saboteur. And at the bottom here, you have the sequence, the actual RNA sequence of this gene. I am not sure that most readers of the New York Times would find it interesting, but for anyone who's interested in bioinformatics, having the sequence is, of course, an essential step. And just reading on from the description, this sabotage forces the cell to make more virus proteins and prevents it from assembling antiviral proteins that could stop the virus. In other words, is a protein that helps the virus dominate the cell which it infects. Next example is NSP4. They call it the bubble maker. And NSP4 helps build fluid-filled bubbles within infected cells. And that's a way that the virus is able to replicate inside the cells without being attacked by the cell defenses. Next example is NSP10. They call it genetic camouflage. And it's a way of avoiding, again, the enzymes that the cell has that may attack the virus. So you see the way the journalists are describing this genome is a way that appeals to notions, everyday notions. And I think they did a very good job in this description. It becomes much easier to understand the coronavirus genome by following these descriptions. Finally, we get to the spike, the famous spike protein S. And again, here you have a description, a representation of the 3D structure of the spike protein. And I put here the computer graphics representation to connect the two. One important thing that everybody who works with coronavirus should know by now is that the spike protein is able to attach to the ACE2 protein, which is the angiotensin converting enzyme. This protein shows up on the surface of many cells in the human body. And because it's there and the virus is able to attach to it, then it makes it much easier. It makes the virus much more infective. It's one of the explanations of why coronavirus was able, or SARS-CoV-2 was able to infect so many people so quickly. And here you have a micrograph showing a cell line. It's not a cell from the human body, but it's a cell in a petri dish infected with coronavirus. Coronavirus are those little yellow dots there, and the cell is the red, these red blobs, and you can see that it's all over the place. And basically, what we are seeing is the spike protein attached to the ACE2 receptor. Okay, I don't want to leave you with the impression that we know everything about there is to know about the coronavirus genome. So an example is this gene here, number 8, of the accessory proteins, the structural proteins. And the description that the journalists gave is that this is the mystery protein. They mentioned that it's dramatically different in SARS-CoV-2 than in other coronaviruses, and we don't know what it does. And so maybe it has some clue as to why the coronavirus behaves the way it does, an additional clue as to why it behaves the way it does. Okay, I'm now nearing the end of the talk. I just want to mention some additional websites. There's the GISAID website, which also is a place where people can do especially epidemiology of coronavirus. And they provide also lots of genome sequences from coronavirus. There's this other site called NextStrain, which uses data provided by GISAID to do tracking of the pathogen. And the kind of tracking that they do is shown here in this map. And this is a screenshot that I took from the NextStrain website. And it shows you the result of comparing coronavirus sequences from different parts of the world. And so you can see here the US on the left, Europe on the right, South America at the bottom. And you have these circles. And the circles, they depict the number of sequences that have been sequenced from that country. And then the lines that connect the circles, they show genomic similarity. So there's this line here connecting the US to Brazil. It simply shows that there was a coronavirus sequence that was isolated in Brazil, which is very similar to a sequence that was generated in the US. And this is a very powerful instrument to understand genetic epidemiology. Was this link because of someone who was infected from coronavirus in the US who traveled to Brazil, and then in Brazil it infected people in Brazil? That could be an explanation. But I think the main point I want to make here is that behind the graph, this map that you're seeing, what took place is a sequence alignment, which is a very basic operation that people do in bioinformatics. And it can be a very powerful instrument. So here you have a more recent map showing connections between Colombia, Brazil, Chile, Uruguay, and Argentina. And it's similar to the other one. Behind this, again, you have genome alignments. But I want to make sure in researching to give this talk, I came across a statement from GISAID, which I thought was important to relate to you. And the statement, I simplified it, but it basically says the following. The first German sample from Bavaria initially looked like the ancestor of most subsequent European cases, because it was the first one, and then it was similar to other ones that came later. But only until more sequences came in from China and the Netherlands, it was clear that the very same variant had been present in multiple locations at the same time. And this means that the transmission routes are much more complex than the early incomplete picture suggested. So I wanted to present you with this caveat so that you do not interpret these maps that I'm showing here that we can conclude without additional investigation that people from country X traveling to country Y brought the infection of coronavirus. This infection routes, as the statement says, are much more complex than what a simple genome alignment might suggest. Okay, so is all of this helping with the pandemics? Well, here, you know, as I said, I am not a coronavirus researcher, and therefore I can only follow the news as everyone else, but I want to leave you with at least one example, which I thought was important. And this is a paper which was uploaded to bioRxiv about a month ago or so, so very recent. And its title basically says everything. Genomic determinants of pathogenicity in SARS-CoV-2 and other human coronaviruses. And what the authors have done is that they used integrated comparative genomics and machine learning techniques to identify key genomic features that differentiate SARS-CoV-2 and the viruses behind other coronavirus outbreaks from the less pathogenic coronaviruses. In other words, this is computational analysis. In other words, the authors here, they have used bioinformatics to, based on the genomes that are available, to infer new knowledge about coronaviruses. And this is a figure from the paper. I want to go. I don't have time to go through the details of this figure, but just very quickly, you can see that there's a phylogenetic tree on the left side. Here you have a multiple alignment. Here you have 3D representation of the proteins that they found more interesting, and so on and so forth. So I think this is an excellent example of how important bioinformatics is for the extraction of knowledge from genomic sequences. Okay, with that, I reach the end of the talk. I hope to have convinced you that genomics is an essential tool for the deep molecular understanding of all life forms, and that genomics is made possible in part thanks to bioinformatics. I also wanted to let you know that we will host the Brazilian Symposium on Bioinformatics 2020, November 23 to 27 this year. This is going to be an online meeting. In previous years, it has been an actual conference, as nearly all conferences, but because of the pandemic, it's going to be online, and the coronavirus, the call for papers will be out soon. And I thought that this community would be interested in learning about that. And with that, I finish the talk. You have my email address there if you want to write me asking questions, or if you're interested in my research group. And I thank you again for the invitation. I thank you for the attention, and gracias por todo.
Okay, thank you, Dr. Setubal. Thank you, everyone. If anyone has any questions. Okay, so there is one question here. It said, how much can we rely on functional inference just by carrying out in silico experiments?
The answer to that question is that in silico experiments or any computational analysis, no matter how powerful they can be, they will only generate hypotheses. Sometimes these are going to be very strong hypotheses. Sometimes they are going to be weak hypotheses, one way or the other. In order to be sure about the prediction or the explanation, you need to do experimental work. And therefore, bioinformatics, no matter how complex, how sophisticated, it's, in a certain sense, can be seen as one step in a chain. And at the end of this chain, or at one of the ends of this chain, you need experimental work. So by this answer, I'm not trying to decrease the importance of bioinformatics. After all, I am a bioinformatician, so come on. I will not say bad things about bioinformatics, but I have to recognize that in order for you to, for example, obtain a vaccine or an effective treatment, experimental work is essential. But the key point is that bioinformatics makes experimental work much, much easier. Because if you didn't have experimental work, the number of alternatives, the number of hypotheses, the number of strains that you would have to look at, the number of cases would be astronomical, and nobody would do that. And therefore, without bioinformatics, biology, especially molecular biology, would have to go back to what it was 50 years ago, which was small-scale experiments. And bioinformatics made possible large-scale experiments, or better expressed, experiments that are much more targeted, that have a much higher chance of succeeding.
Okay, thank you. So there is another question. So Juan Martinez, he asked, what's going on? So if we compare like the number of genomes that are being sequenced by the US or Europe, compared with the number of genomes that have been sequenced by South America, why do you think that we in South America have just a few coronavirus genomes? So do we need like a technology? Do we need the experts that analyze those sequences? What's going on there?
I think lots of things are going on. I can't speak for countries that are not Brazil. That's where I live, I'm most familiar with. And so I would rather answer the question in the context of Brazil, and then maybe, every one of you could see if the explanations I'm going to give generalize to other countries in South America. So the first thing is that in order to generate a complete sequence of a coronavirus, a genome sequence of a coronavirus, many steps have to be accomplished. You need, and in each of those steps, you need people with expertise and you need money, right? And so if there's a lack of people with expertise, if there's a lack of the infrastructure, if there's a lack of money, then the end result is going to be a fewer number of sequences generated. And not only that, but those labs in Europe and in the US that are being able to sequence all those genomes, they have procedures in place that make it the sequencing a much more routine work than it is the case here in Brazil and perhaps in South America in general, right? In other words, I'll try a comparison here, a metaphor. It's like a factory, right? So you have a factory in Japan, for example, that can produce 1,000 cars a month. Whereas here in Brazil, we don't have a factory like they do with robots and so on. And therefore, the production of the car factory here, instead of being 1,000 cars a month, it's going to be two cars a month. They are able to produce 500 more cars every month than we can. But that's because, they have the robots, they have, the expertise. It's not that we cannot do, we are able to do it, but they are able to do it in a much more efficient way. And they have more people and so on and so forth. So I think I'll stop there because if I wanted to give more details, then it would be a much more technical answer, and maybe that's not what's needed here.
Yeah. Okay, so two more questions. So Paola Aguirre, she asks, how many coronavirus sequences do we need to align around the world to determine the number of total variants, or what determines that a sequence is a new variant?
Well, the first part of the questions, how many coronavirus sequences need to be sequenced to determine the total number of variants? The honest answer is infinitely many, because this virus, as all viruses, it's still evolving. It's changing as it spreads. And every time the coronavirus changes because, of random mutations, because of adaptation to new environments, the sequence changes and it is a new variant. And therefore, potentially this number, it's not infinite because it's not infinite because if the virus has 30,000 base pairs, then 30,000, 4 to the 30,000 is the maximum number of variants that you would have. Right, but this is a mathematical abstraction. It's meaningless in the real world. In the real world, it behaves as if there is an infinite number of variants, which means that people will have to continue sequencing and see what's going on with the virus, if it's still evolving and how fast it is evolving and so on and so forth, right? As to the second part of the question, which I forgot, I was reading the question here. Let's see. I can see the chat here. Let me go back. What determines that a sequence is a new variant? I think the basic distinction here is the distinction between a sequencing error and a sequencing or a sequence difference that is real. It's an actual mutation. Because sequencing is not perfect, every time you sequence a genome, even a small genome such as a virus, you will have sequencing errors. And you have to be careful when you're comparing different sequences from different genomes, not to mix up what is a sequencing error with a true mutation. So once you have determined that a sequence difference, a genome sequence difference is not an artifact, then even if it's just one single mutation, in principle, you could call that a variant. But I would say that just one is too little, right? You need more examples. And how many, I'm not sure. but I will guess that virologists will look at a given genome sequence and say, well, in order to call that a variant, I need to have seen a certain minimum number of differences. Let's assume like something like 10 differences, 10 base pair differences scattered throughout the genome. And I need to see those 10 differences represented at least, I don't know, 5 times. 5 times meaning 5 different isolates. Meaning this isolate came from person A, this other isolate came from person B, and they were sequenced by different laboratories. And okay, once I have this, I can call that a variant. That's my guess, but I need to say that I'm not totally sure that the numbers I gave you are the ones that are actually used in practice. But if they're not the ones that people use, they shouldn't be too far off, right? The essential message here being that just one mutation does not characterize a variant. But on the other hand, you don't need 1,000 mutations to characterize a variant.
Okay. So Juan Valdez, he said, if there any like tool for genome assembly that is specific for viruses?
Yes. There are several assembly tools available. And I don't have a list on the top of my head here, but one very popular is called SPADES. And SPADES is an assembly tool that has been used in my lab. For example, we used to assemble bacterial genomes, but we have also used it to assemble viral genomes. And so I would, in part, say that SPADES is a very good tool for genome assembly, and it should work well with viruses. But very recently, the same group that produced the SPADES software produced a variant of SPADES that they called, I think, called metaviral SPADES. I'm not sure about the name. And I believe that version is a version of SPADES which aims to assemble viral genomes from metagenomic data, which is not exactly what you have asked, right? But I think this is an example. I think I'm using this metaviral SPADES new release simply to emphasize that SPADES, the general SPADES program is a very good program for assembling viral genomes.
Okay, so we have maybe just the last question. So David Requena, he asked, the current coronavirus came out of a reservoir noticed by the scientific community long ago, reservoir that did not receive the proper attention. As Brazil has a large diversity of species, have you heard about any animal disease with reported potential of crossing the species barrier to infect humans?
That's a very good question. And I think the quickest answer that comes to my mind is the yellow fever virus. So yellow fever is a recurring problem here in Brazil. And it is known, and it has been known for a long time. It's not new knowledge. It has been known for decades, or even more than decades, that yellow fever virus can jump from monkeys to humans and back to monkeys. And so, and because of that, whenever there is a yellow fever outbreak, there's always the concern that either it may have come from contact between people and monkeys, or the monkeys themselves may be threatened by yellow fever from humans. So that's the best example I have. As I said, it's been known for many, many years. But I think the question perhaps is aiming at the possibility that some new virus might come up from reservoirs in Brazil in a way similar to what happened in China, right? The information I have read about the outbreak suggests that the coronavirus jumped from wildlife in China, from those markets that trade with bat meat or pangolin, that might have jumped to humans. And so I think the question is, could that happen in Brazil? Maybe there are some wild animals being trafficked in the Amazon and they have some unknown virus, and there's this market in this little town in the middle of the Amazon, and suddenly a virus from there jumps to a human and then spreads from there. Well, I'm not a virologist, and even less coronavirologist, and so all I can answer is that this possibility certainly exists, and I know that there are researchers that are concerned with that, and I know that there are research projects that are going on with the aim of finding out more about these reservoirs. And I think this was already taking place before the current pandemic. And I think Brazilian authorities and Brazilian research agencies will pay even more attention to this possibility than they did before. But how much of a risk this is for the future, I cannot say. Right? I don't know. Maybe what happened in China was something extraordinary. Maybe not. Maybe there will be new pandemics in the future, either from China or from Brazil or from Africa. But not being a virologist, I don't feel comfortable in answering in any more detail than I already have.
Okay, we want to thank you, everyone, for being here today, and also this Peruvian Society of Bioinformatics wants to thank you, Dr. Setubal, for this opportunity and for your time and for the time that you are sharing with us here today. And we want to invite everyone else to for June 5th on Friday at 6 p.m. Again, we will have a new seminar. And yeah, so we want to thank you, and we hope that every one of us, or if anyone has any questions, maybe they can just send you an email or contact you, and or if you want to say something to everyone else before we finish this presentation.
Yes, I was going to say that I can see in the chat here many of the questions that people have asked, and I think I've answered only a few of them. So I would have a request to you, is that to save this chat and send the questions to me, because it's important to me to know what people have asked.
Okay, yep.
I'm not sure I'll be able to answer them all individually, but to have the questions, it would be important to me. So I would ask you to save the questions and send them by email to me. And if anyone really wants to ask me the question that wasn't answered, please feel free to write me. If I get emails from people who have attended this talk with questions, I will be more than happy to answer all of them.
Okay. Yeah, thank you once again, and we hope that we want to be in touch with you. Maybe that we can talk, or we can collaborate with the Peruvian Society of Bioinformatics in the future, and see how we can try to come up with new projects, maybe in collaboration with Peru and Brazil in the future.
Okay, that would be excellent. You can count on me, Marco.
Okay, thank you very much. Thank you. Thank you, everyone.
Nada, hasta la vista.
Luego. Hasta luego.
Thank you.